You know, guys, there's this commonly asked question in your histology exams. Many students generally get confused with this. That what is the magnification of a microscope and what is the resolving power of a microscope? So this is a question to Swami. Swami, tell me what is magnification of a microscope and what's the difference between resolution of a microscope?
Magnification is the magnification to increase magnification of the objective lens.
Objective lens. That is how bigger the size is going to be. That's just increase in size. Magnification is the enlargement of
Actually, it is enlargement of the field without, adding to your knowledge as far as the details are concerned in the tissue, without increasing. The same knowledge, say there is a cell, there is a nucleus. You just increase the size 1,000 times. You will see only a cell and a nucleus. Resolving power, it makes you able to see more details in the tissue. And the exact definition is, see, minimum distance between the 2 points which are seen as 2. I will give the example your own eye. The resolving power of the eye, is approximately. It is 0.2 millimeter. 0.2 millimeter. What do you mean by 0.2 millimeter means? 1 millimeter divided into 5 parts. Right? Then every line between about will be 0.2 millimeter from each other. So that is the maximum you can see with your eye. But if you divide them into 6 parts, 7 parts, then you cannot see, right, the 2 lines. It will look like a belt, dark belt. But the point, the same thing is true here. Right? There, for example, resolving power is 0.2 micrometer per day. Right? Usually the scale we are using for eye, centimeter, meter. For microscope we use it, usually in micron. For electron microscopy it is nanometer, the standard. Right? Angstrom was used earlier, but now it is nanometer. Right? So that the measures which come, they are in the range of 0 to 500 and 1000. It is not 1 lakh or 2 lakh. You got the point? So that is the actually resolving power. So there is a formula for, I will not go to detail, to calculate the resolving power. Dr. Azmi has already told you how to calculate the magnification. Magnification, the eyepiece is 10 times magnifying. Objective 10 times, you just multiply them. Total magnification will be 100 times. But without, making any detail, as far as the structure. But if the resolving power is good, right? For example, in electron microscope, you can see all the 3 layers in the plasma membrane, right? Because the distance between them is 25 micron. Right? Total thickness is 75 micron, for example. So resolving power is 3 to 4 micron, for example. So you can see all the 3 lines. That is the details. Right? So more important, is resolving power. Right? So this is very important, for especially for you post-graduate students. Right? Because you have to become teacher, you have to explain to the student why we are using electron microscope, why we are using. Did you get the point?
So, all you guys, you must be taking good photographs from your mobile cameras these days. And when you see the images, you span, zoom out like this. The images, the more you zoom out, the images become blur, right? Although you're zooming out. So, that zooming out of an image is a magnification thing. But the more the megapixel, you're getting the market of 13 megapixel, 18 megapixel. So, the megapixel is like increased in those, the megabytes of those pictures. The density of the picture is increased, like you using high density images. So that is compared to the resolution power of a microscope. Resolving power means increase in the density of that image what you captured, so that you can easily appreciate the minor details even after zooming it out, right?